Bone marrow stromal cells are an important component of microenvironment. It's known fact. The stromal cells secrete a wide range of cytokine, growth factor, and matrix protein that stimulate the adhesion of myeloma cells to the bone marrow, protecting the cells from chemotherapy and helping to accumulate myeloma cells in bone marrow. The tumor microenvironment activates multiple pathways leading to the lesion growth and disease progression. And in fact, all biological processes active in microenvironment can contribute to myeloma progression and development drug resistance. We can say that bone marrow stromal cells variation are initially induced by tumor plasma cells, but later these cells create favorable microenvironment that support proliferation, migration, survival, and drug resistance of myeloma cells. And based on this knowledge, we contribute our study of stromal cells obtained by long-term cultivation from myeloma patient. First, we analyzed global RNA expression in bone marrow stromal cells from myeloma patient with active and non-active disease. I mean, for example, complete remission and progression of disease. In comparative analysis of comparative expression analysis revealed more than 70 genes. Those expression differed significantly between these two groups of patients. After appropriate statistical and bioinformatical analysis, we determined that most of these genes are involved in acute inflammatory response and intracellular trafficking. So we also analyzed then expression of profiling expression in stromal cells from individual patient in different stage of myeloma, for example, at diagnosis, progression, complete remission, some late years later. And we found significant changes, especially in the interleukin and related cytokine pathways. Then next we analyze effects of co-cultivation in bone marrow stromal cells and myeloma on gene expression, but our previous data supports this data. And in the end, finally, we tested several drugs, Lenalidomide, Elotuzumab, and Daratumumab in co-culture of bone marrow stromal cells and myeloma cells. After adding Elotuzumab and Lenalidomide in co-culture of stromal cells and myeloma, we found significant changes in the expression of genes for the interleukin pathway and hematoxis pathway. Interesting that especially strong changes we have seen after lenalidomide treatment. The addition of elotuzumab always reduced the effect of lenalidomide. It is a very interesting observation and very good correlated clinical situation. In Daratumumab treated co-culture, we discovered not a lot of genes, but at least 11 genes, which were significantly differently expressed in stromal cells. And especially integrity pathway analysis of the genes reveals that most of overrepresented canonic pathway were the interleukin 17, CD40, and NF-κB signaling pathway. So we can say that our data confirm that interaction between myeloma cells and stromal cells is a very really complex mechanism based in multiple interaction signaling pathway. And this interaction increases through the course of disease and may contribute to the development of drug resistance. This is a fact that microenvironment supports tumor progression, and we think that's currently drug or new drug must not only act on tumor cells, but also counteract the tumor promoting effect of tumor environment.